Dear students, in this module, I'm going to discuss the chemical composition of RNA. RNA or ribonucleic acids. This is another major type of nucleic acid present in the cells. It is also a polymeric compound, just like DNA. As you know, DNA is made up of deoxyribonucleotides, but here RNA is made up of ribonucleotides. So ribonucleotide is the monomer of ribonucleic acids. So each ribonucleotide is made up of three components, and these three components include a ribose sugar, a nitrogenous base, and a phosphoric acid. So these three components, they join together and they make a ribonucleotide. And different ribonucleotides, they join together and they make a polynucleotide, which is ribonucleic acid. Here, this is the first component of ribonucleic acid, and this is a pentose sugar. Here, the pentose sugar is a ribose. You know, in DNA, this sugar is also present, but this is present in its derivatized form, and that form is deoxyribose sugar. You can see at carbon number 2, an OH group is present here, but in case of deoxyribose sugar, only hydrogen was present, oxygen was removed. So here the pentose sugar is ribose instead of deoxyribose. This is first component of our ribonucleic acid. The second component of ribonucleic acid is the nitrogenous base. There are mainly two types of nitrogenous bases present in the ribonucleic acids, and these are purines and pyrimidines. Two types of purine bases, that is adenine and guanine, are present in the ribonucleotide. These two bases, adenine and guanine, were also present in the deoxyribonucleotide. The next two types of bases, that is pyrimidines, and these are cytosine and uracil. Here you can see a difference. In case of DNA, uracil is not present, and instead thymine was present in DNA molecule. But here, instead of thymine, uracil is present. So overall, there are 5 types of nitrogenous bases. These are adenine, guanine, cytosine, thymine, and uracil. Thymine is only present in DNA molecules, while uracil is only present in RNA molecules. The third component of a ribonucleotide is a phosphoric acid. Phosphoric acid, you can see it has 3 hydroxyl groups, and these hydroxyl groups, they have ability to make ester linkages with the hydroxyl groups present on the pentose sugar, either ribose or deoxyribose. So ideally it can make 3 ester linkages, but normally in a polynucleotide chain, this phosphoric acid makes 2 ester linkages. So this is a complete ribonucleotide. It consists of all the 3 components, that is a nitrogenous base, a pentose sugar, and a phosphoric acid. These 3 components are joined together and they make a ribonucleotide, and this ribonucleotide, by combining with other ribonucleotides, it make a ribonucleic acid.